I'm a great pleasure to be here with you. Welcome, everybody. We will start the fourth webinar of International Society of Health Education about last updates on COVID-19. May I ask Dr. Salman Al-Rawaf, the Chairman of Society, to start the webinar by welcome letter.
Thank you very much indeed, Professor Maytham, and welcome everyone, and festive seasons, Merry Christmas, and Happy New Year to everyone. I'm delighted to be with you on the fourth session of the society. The subject we are discussing today is really a very vital subject. The new mutation of the virus, which these mutations are expected in all type of coronavirus. For example, the coronavirus, which is causing infection, actually is changing almost every year. And that's why we are changing the vaccine every year. And we're responding to it in different way every year. So that mutation expected. And since the virus was first diagnosed in China, in Wuhan, in January early this year, we have at least 17 mutations. So let us listen to colleagues today and take it from the real sources of practitioners about what does it mean, this mutation and the new strain which appear in England, which now actually spread across the UK and across the world. And also another new strain of the virus following the mutation, a new variant appear in South Africa and also spreading. I look forward to listen to my colleagues and enlightened by their wisdom and perhaps generate some discussion among all our colleagues. And please add from your knowledge, information, and experience you have, because we are until today trying to find our ways on the best way to tackle the new variant of the virus. Thank you.
Okay, thank you so much, Dr. Salman. Now, Dr. Khalid Shafi, he will talk about the new strain of COVID-19. Welcome, Dr. Khalid.
Well, thank you very much for inviting me, and thank you to Professor Salman, Professor Haytham, and all my senior colleagues in the panel. I wish everybody a happy festive season and happy New Year. And let us hope that the new year, which is 2021, will bring us a lot of hope. Now, the issue about the variant CJD, next slide, please. There is a famous saying in English. It says that if you have been bitten by a dog, the name of the dog is not important. Is it true? Maybe true to some extent, and maybe wrong, because science developed very much. So we need to identify the organism and to identify its epidemiology, and as well as we need to get help from this identification about the pathophysiology as well as treatment and vaccination. This argument may apply to somebody who has been bitten by a dog in the UK, and we say to him, don't worry because we don't have rabid dog or all of them vaccinated. But about other organism, we must know the organism, and with this will help us actually to tackle the issue. Next, please. Now, why we have a new COVID? Is it something that unexpected? And what does it mean? Is it natural? What we are going to do about it? And if we are already infected, are we going to be reinfected with it? How the current vaccine are protective? And can we get some assurances? These are the questions that everybody asks about them. And science, already there are a lot of science professors, laboratories, physicians, they always work hand in hand and exchange information regarding these questions and their answers. Because as you know, research and development depends on the good question that need to be answered. Next, please. Next slide. Now, the ecosystem. Most of the people who have less information about microorganisms or microbiology, infectious diseases, think that we are living in a sterile world. This is absolutely not true. We have an ecosystem full of microorganisms, living organisms, animals, human beings, parasites. And this is helpful because the world is actually created by the Creator is to help each other. So we need the bees, and there was an issue about the insecticides in parts of America that killed the bees, and fertilization of the crops actually was a serious problem. This is just an example. So we are living in a place that actually we need each other. The symbiotic interaction between commensal organism and human being are essential to develop the immune system, as well as to protect us and sometimes give us essential vitamins, as well as other, protective materials like fatty acid on the skin. So infectious diseases and microbial diseases need to be understood in a different way from other diseases, because it's an interaction between two biological systems. And we need to know that, actually, because this is the issue, why we get resistance to antibiotics, why we get immune escape, like, hepatitis B, and why we get some people infected. Some people go to the hospital, some people stay a few days at home, some people go to the hospital, then to intensive care and die. What do we mean by immunity? And what do we mean by immunocompromised situation? And these are actually big fields. Still, we are swimming through them. Now, the other thing about microbial diseases, there is what we call cross-infection. Cross-infection will create outbreaks, clusters, epidemics, pandemics. Hence, COVID-19 is not an exception. Next, please. Next slide. Now, how we survive all these years, and can human being or mankind change to another form? Can we, as a human being, to protect us, borrow or acquire another gene? Can we modify our genetic expression? The last question may be because we are not, or we cannot actually acquire a new gene, because human being is made of highly specialized tissue. Only at the embryonic stage, they can actually modify, or we can treat, or they can affect it to a serious or a major extent. But as we grow over, we may modify gene expression, and this can be in the form of immunity. And if I remember rightly, this is you remember that the new variant CJD, which is the only second infectious agent, although there's a debate whether the new variant CJD is an infectious organism or actually a transmissible protein. This is the interaction between us and the environment is completely different when we go to the microorganism, which is the simplest form of life. So when we interact with the environment, and here I'm concentrating on the microorganism, we get diseases. And this is a natural phenomenon. Then mankind actually is much clever creature, try to make vaccine, to make medicine, and have some behavior changes. Hence, infection control is mainly a behavior science. So this is how we adapt to the environment, by our gene expression and also by intervention, which I mean by creating vaccine as well as medicines. Next, please. Now, the microorganism. The microorganism, and as you go, the smallest microorganism, the easiest for it to mutate, to acquire new genes. And in fact, this phenomena, we as a human being and in research, we actually benefit a lot to study the genes. And hence, the vaccines, the vaccines actually borrow ideas from the mutation. Because when you take a gene, you take its function, and the function will express as a phenotype. So there is a genotype and phenotype. So resistance to antibiotics is based on gene expression as well as actually borrowing genes. And in bacteriology, it's very well known that was more than 30 years ago, you get the transposon, plasmid, and in addition to the mutation. And then the microorganism, by borrowing genes, they modify their targets. And I'm talking about the target of the antibiotic, target of the antiparasitic, target of the antiviral. So the microorganism has to survive and has to adapt to the environment by borrowing more genes. It hasn't got the complicated brain of a human being, but it is, again, it has some way of protecting itself to survive and, continue in life. So life is about interaction and survival of the fittest. Now, the organism also has the capability of evading the immune system by, simply bacteria make abscess and or stay dormant like cysts or spores, and hence in these forms the antibiotics has no or minimal effect, or they can inhibit the immune system, which is explained by the phagocytes. Example, we have a lot of example, from herpes virus, herpes simplex, and chickenpox. They had somewhere, and then they produce themselves again. TB is an example. So when we talk about TB, actually there is no complete cure. TB will come back to you again when the immune system, weakened. Similarly, hepatitis B. And this is very important when it comes to transplantation. So still, we are in a battle and we are in a balance, and always there's a balance between the immune system as well as the microorganism and the whole process called adaptation. Next, please. Now, how to deal with a change or possible change in the microorganism? And this is actually, we have tremendous experience, knowledge, and equipment, and infrastructure. It's by improving the laboratory detection of the microorganism, as well as the biomarkers. And there is a bank for biomarkers which is established more than 10 years, which is studying all the changes in human being. including the CRP, which is the simplest example, procalcitonin. And they are taking these samples and storing it, and to see in reference to the future, to see whether we got any herald signs of an impending serious disease, in addition to the diagnostic capabilities. And, the diagnostic capability now much more advanced into a molecular based, and this is not, based on a single stranded, RNA or DNA. It's what they call it multiplex. And that is why, how we discover COVID-19, the new variant, because we have multiple DNA strands that detect any changes in the virus and will tell us. And we have they call it biobank or gene bank, and we have a reference to it. And hence the diagnostic process will discover a lot of diseases. In fact, if you take example of hepatitis C, it is not culturable virus, but has detected by the molecular detection system. And the other thing is the increase in or improvement of the clinical acumen. This is, based on the university experience, expertise, literature review, guidelines, cross help between, you can now see a lot of articles, through the electronic system, which is very helpful. I mean, each one of us will, if I had a different case, you look into in Google or any Medline or anything, then that will help to exchange expertise. And one more about the biobank, because in infection and microbial diseases, there is a certain limit to what you can do, and what they call it a threshold. And beyond that threshold, we lose control on the disease. And this is very well known. When I go to the ITU as part of my job, the patient got antibiotics or antiviral, but reach a stage when the cytokine storm started, nobody can stop it. It depends on the immune system of the patient. And hence, COVID main picture is due to cytokine storm. And that is why they use anti-inflammatory, and the main one is steroid. So, which is first? So go back to the first slide, the previous one, please. No, go back. So we need to detect the cause, and the second, detect the tribe of the organism by serotyping, genotyping, where it comes from, the ancestor, and I've got a slide about the phylogenetic. And this, they talk about the detection and tracing. It is very important. You have a baseline, you have an infrastructure about the detection process. And COVID variant is an example, and it is testing our test and trace system. Next slide. Next, please. Now, COVID-19 variant, this is the lineage description. In fact, the updated literature saying that it has 20 mutations, not 17. I just read an article, and it is belong to the B lineage. And when we talk about lineage or phylogenetic, I mean, they are so far, because this is an evolving situation, so far the lineage or the ancestor is A, which is very much linked to Wuhan, and then C belongs, or mainly in Europe, and B, Southeast Asia. I don't remember exactly where. And B, it has to have a lot of mutations to reach Europe. Now, we got the variant, which belong to lineage B, and this is 1.17. And this is, when you look at the genetic tree, it's just like a family tree. You get the parents, and then as you go with time, this will have more modification, and hence you get various serotype or genotypes. Now, detection is by multiplex, I've said that. And then why we have a new variant? Why the virus mutate? Because of the selective pressure. This is maybe to natural antibodies, or it has been suggested now recently that the plasma may be given to some immunocompromised, and because they are having the virus for a longer time, which is very well known actually, we learn that viruses can stay longer in the body when the immune system is lower, and this is the example is ITU patient and swine flu, and the other example HIV patient, which has a lot of potential of mutation. So it could well be, some of the literature say that probably the plasma given to some immunocompromised patient, and because of a lengthy period, you get mutation. And this is very well in bacteriology, because in bacteriology, when you have a mutation, especially when you test antibiotics in the test tube against an organism, there is one of the parameter, you can say what is the actually chances or the frequency of mutation, and the good antibiotic will produce less mutation, because it's a selection process which is very well known in antibiotic resistance. So why it is going to be the dominant strain? Again, it's a survival of the fittest, and it has, as has been explained, a high efficiency binding to the cell, because there's a modification mainly in the S protein or the spike protein. Now, is this adaptation is a serious thing? So far, there is no evidence that it is this new variant, although it is adaptable, is going to be more virulent, i.e. more nasty, more causing disease. It could be just, an adapted strain. In fact, we have the same example with swine flu. Initially, there was a lot of media interest. People get swine flu, died, and I've seen them in the intensive care. Then, after a while, swine flu became a seasonal virus. And by the way, the mutation in SARS is highly frequent. In one of the studies saying that each month you have 2 mutations. And so far, these are minor mutations. It has no effect on the severity of the disease. And the other point is that, in fact, so far, I must say, in one of the literature says, so far we had about 1,200 mutations, but all these minor. The worry about this mutation or this mutant or variant, whatever you call it, it has an aggregate of about now 20 mutations. So far, there is no evidence about saying, well, it has more infectivity to the children. It may be, it has no virulent effect or increased virulence in children, but may be more adaptable, and maybe the children is more infected. And I must say, infection is different from death or morbidity. We may be happy to get all the people infected, and hence we get the herd immunity, and this is the function of the vaccine. So, but the worry is that people are watching the situation, and so far there is nothing to cause any concern. Now, do we need to do more? Are we scared? Is it more virulent, more transmissible, or more adaptable? I think I've answered most of these questions, but there's a a terminology that has not been used precisely. When we say that it is 70% more transmissible, in fact, if you look at the virologists or the data, this new variant started or detected in Kent and then spread to southeast England and now more in UK, and more actually similar mutant appeared in Australia and part of Europe. It seems that the virus or the new virus is taken over from the rest of the circulating COVID, if you like, serotype or genotypes. It was in September 25%, now changed within a short period of time to 70%, which means it is the dominant genotype. Now, it is different when we say this is more transmissible. Transmissibility is probably a difficult thing to measure. However, the PHE guideline said that it is enough for somebody to be cross-infected from an infected person if he is sitting face to face within a meter without face covering. And this is a measurement of transmissibility. So we do not have these data for a new variant, although the terminology is used very loosely. Next, please. Well, this is a slide, I borrowed it, and it shows the adaptation process and the ancestor, and then it goes down to the species, and all this, how we know this is the ancestor, because in genetic typing, and especially in PCR, there is called a conserved region and a variable region. So the PCR should detect both the conserved region, which is like a trunk of a tree, and the branches are the changing part. So if the viruses are similar in terms of the trunk, we say this is a COVID-19. Then if there is variation in the branches, then we say this is a new variant. And then, the new strain come up as we go along, because this is because of adaptation. And I must say, before I forget, during the swine flu, there was still bird flu, which is very, very serious and virulent with a high death rate, H5N1. But so far, it transmitted, as an occupational disease from the bird, chicken to the human. It's a deadly virus. However, there is no human-to-human transmission, which so far we are lucky. But once there is adaptation, we may expect, an epidemic or pandemic, and then it will die down like any other pandemic. So this is sort of evolution and adaptation. Next slide. Next slide, please. So the answer to all these questions require improved and continuous laboratory detection and exchange of information between laboratories. We have a good system now between the States, now China, Europe, and UK. Whenever you say there's a guidelines, you find that the guidelines, at least in 3 or 4 places, and one actually complementary to the other. And then this is a good thing. Continuous clinical vigilance in terms of identifying, the new clinical picture, because sometimes a new clinical picture may tell you there is something new. And as Professor Salman said, now most of the new infection, actually, they are less pulmonary type, more of the fatigue type, change of smell, mild fever. And this is probably a good sign. Improve other biomarkers and vital organs function test, and see whether it is more virulent. during my experience as an infection control doctor, when there is an outbreak, we sometimes close the ward or 2 wards. The first question we ask, is there any patient died, or anybody has been taken to the intensive care, or any bleeding? Because these are very important when you assess any virulence, assess any change, and give you a stimulus to act, urgently. But nowadays we are more sophisticated because the laboratory and the detection is so advanced that we now tell or say or share that this is a new variant. Guys, please, be alerted. We got a change in the virus, and this is really, really useful and important. Now, good infection control, namely test and trace. This is really very important and still working, and it should continue. But I must elaborate on the testing and tracing. There are successful countries in this test and trace, namely South Korea, Vietnam, and 1 or 2 other countries. They use the epidemiological data, and they go back, the contact of the contact. And they go back against before the contact. Where was this person? And they gather the data. And by gathering the data about the contact, as well as examining the virus genetic composition, we can even trace any new virus. Just like when we were, doing this for Salmonella 30 years ago. Now, monitoring effective vaccination. And as I said initially, so far there is no evidence that the new variant can escape the vaccination. However, people are just watching the situation because if the mutation becomes so much that it can evade the vaccination, then this can be a problem. But this is, everybody is watching it. And remember that when we are vaccinating, it's not, although the vaccination is mainly on the spike proteins, and the spike protein has been shown to change in this virus, but the vaccination as well as the immune system is so robust and complicated, produce a lot of antibody, not only antibody against the particular type of spike, but there are other associated or what you call it, cross-reacting antibodies, if you like, and that give us a wider protection just in case the virus change its structure, which is why human body is so sophisticated machine that it has the protection and the expression and the adaptation. Now, next,
Dr. Khaled, you have just 5 minutes to finish your lecture, please.
Okay. I think I'm nearly done now. Okay. Does lockdown, Tier 4, and restriction of movement reduce the takeover of this new variant? The answer may be, but it may end up being more, the lockdown can be disruptive, which we know that, or counterproductive. However, the natural selection phenomena, which is the adaptation of the virus, could well give us more explanation. I hope is not going to defeat the lockdown, because the presence of this virus in different parts of the world may mean that it is a natural mutation anyway. It has nothing to do with people traveling from London or Southeast. And let us concentrate more on the infection control, hand, face, and space, and test and trace, and monitor the vaccination program. Next. Thank you very much for your attention. Any question?
Thank you so much, Dr. Khalid.
Is it possible to comment and ask questions?
Excuse me, Dr. Majid. After finish Dr. Sima, you can discuss or you ask Dr. Khalid. Okay. Now, Dr. Sima Karantinaya, she will talk about can vaccine be a cause for a new strain generation? Okay.
Yes. Yes, good afternoon. It is afternoon in the United States. I wish everyone Merry Christmas and happy any upcoming holidays that anyone celebrates. We want to make sure to let people know that public health care issues are issues that are universal, regardless of culture, country, race, religion, gender, any other category. So I would like to wish everyone a healthy and a safe holiday and life beyond the holiday. What I would like to say is that, first of all, I want to make it very clear that many conspiracy theories are arising around the vaccination alone. Basically claiming that the vaccination is used to control the human species, just to control and modify and sterilize, if you will, the human species for the purpose of, as someone says, the puppet master who's going to utilize and instrumentalize the human species in their for their benefit. This is not true. What we are trying to accomplish here is we are trying to get the coronavirus pandemic to die out as quickly as possible. Not with the speed of light, because we know that it's difficult, but as quickly as possible. With that being said, I would like to state that the vaccine developed by Pfizer and Moderna is not the cause for the new strain. It is actually the protection for the new strain. And I am going to go into details with this. There are two different mutations that I found out about. One of them is the N501Y, which it has been found in the South Africa and other geographic areas. This particular mutation has found to have competence towards binding. So basically, it improves the virus's ability to bind to the receptors that allow it to infect our immune cells. And it has been shown through different studies that this particular variation is as close to the original COVID-19 as possible, which basically means we have absolutely nothing to worry about. Now, the D614G variation that has been found in the United States, and unfortunately, it has been found in the United States after the UK has spotted any mutations of the original coronavirus. The reason being is because the United States, unfortunately, does not have that strong of an infection control surveillance program as the UK does. This is what we are working on improving, which is why many people, many professionals and representatives in the healthcare field here in the United States are saying that there is no public healthcare system at all. There is a public healthcare system. It's just that it needs to be up to date. So this is what we're working on. Anyway, back to my topic of discussion. The D614G variation that has been found in the United States is involved in, the spike protein mutation is involved in replication. So it improves the virus's ability to replicate in the upper respiratory tract. What it basically means is that the virus, the new virus variation, is more likely to spread through coughing or sneezing. And as we know, maintaining social distancing and wearing masks can very well control these particular aspects. What is good about the fact that it only replicates in the upper respiratory tract is that there's a very high chance of the new virus not getting to the lungs, which means the patient might have a less likelihood of having acquired pneumonia, pulmonary embolism for those that have cardiovascular issues, in addition to respiratory pulmonary issues. Therefore, these patients have less likelihood of dying because the actual complication, the actual disease process as a complication of this virus is going to be less difficult to manage clinically, pharmacologically, and long-term after the patient gets out of the hospital. So basically what I'm trying to say here is that it has been proven, it has been shown in many studies that the nanotechnological RNA vaccine that has been developed right now is able to treat 90% of all cases of COVID-19, and that involves the the new variation that was found in the United States, which basically means it is our friend and not our enemy. The vaccine is developed quickly because of the pressure on governmental officials and entities, as we discussed in the previous webinar. However, in my opinion and in my prediction, I would say that the vaccine has been developed successfully. The starting point of this new RNA vaccine is very successful, has a very positive outlook, and we can only advance it in the future, which basically means that it is not going to cause new strains of COVID-19. It is only going to allow the pandemic to die out, meaning to disappear, to become extinct quicker, not quickly, once again, not with the speed of light, but quicker than it would have been otherwise. So I would strictly follow my previous colleagues' advice, Professor Khalif. You said it completely right, that we have to monitor infection control. We have to become strong on infection control, especially here in the United States. We have to continue collaborating with all the different countries of the world, like the countries all of you, my fellow colleagues represent, and other countries that are yet to be involved in our society. And monitor infection control, monitor the testing and tracing, and become very, very pivotal thinking and pivotal initializing on the vaccination. And one very last point before I wrap my speech up. It is important to administer 2 doses of the vaccine. It's like an antibiotic course. If you have a disease and you feel in 2 to 3 days after having taken antibiotics that you're totally fine, you stop taking a 7-day course or a month course of antibiotics, there's a chance of you becoming reinfected or having complications that are far worse than the previous infection, which means if we only stick to 1 dose of the vaccine, we're only partially safe, and we want to be fully safe considering what we went through starting actually February of this year. We do not want it to repeat. And once again, we want the pandemic to become extinct as quickly as possible. Thank you very much. And I wish everyone health and all blessings.
Okay, thank you so much, Dr. Sima Karatinaya.
Thank you.
Anyone want to discuss further point with the speakers?
Can I just comment,
yes,
if I may?
Dr. Majid, yes.
Thank you very much. Firstly, I would like to thank both speakers, Khalid and Sima. And Sima, very happy Christmas also. I hope we have not interrupted your, your celebration or whatever. Okay.
No. Thank you.
So, thank you for alluding on 2 important points, both the the new mutation and the vaccination. Just back to what Khalid alluded to on infectivity. I quite agree with him. Infectivity is completely different from morbidity and mortality, you know. And in children, because he mentioned children, okay, you know, in pediatrics, we haven't observed, I'm a pediatrician, by the way, Seema, we have not observed an increase in number of pediatric in symptomatic children. Let's say symptomatic. We don't know whether they were infected or not, because we know the symptomatic children anyway are a very small number, even with the first waves and second, whatever, okay, with whatever variants we had. But with regard to the multi-system inflammatory syndrome, the case definition of which is very well defined with all these severe symptoms and fever and multi-system infection and cardiac-like symptoms, which is Kawasaki-like. It's different. We don't know. We cannot say we haven't seen as yet, because you know very well it followed a month after the peak of the first and second wave. And it was not related to immediate viremia. It was related to immunity dysregulation, which happened a month later in children. And then they presented with all these rather severe symptoms of fever, cardiac symptoms, and shock, etc., toxic shock syndrome, etc. So these points I thought I will just go over on, just to make it clear, especially in pediatric. Thank you very much, both.
Okay.
Thank you. Regarding the long incubation period, as Dr. Majid said, we know the virus evades the immune system, and that is sometimes multiply for a long time in the macrophages, and then it appeared like a cytokine storm. This is very well known. And it is very interesting to know that the anti-spike proteins are very high in severely ill patients, while those with mild disease, they have longer protection. So you got a disease, severe disease, after recovery, they have a short span of protection. Yeah. And those with mild infection, they have a longer span, up to 6 months. As I said, that this is in an evolving situation. Now, the other thing about if you say there is no evidence, it doesn't mean the absence of evidence, because we are learning again. But there are three terminology. It's the infectivity, the severity, and transmissibility. Yeah. And the transmissibility is a loose term. Use it, and they mix it with transmission, which it means what they are saying, that the new strain is actually taking over and comprising 70%. Now, the vaccine, it may has an effect, because in our previous knowledge, say from meningitis B, after we vaccinate against C and A and W and the other types, we noticed there is a shift, an increase in meningitis B. And so there is a lot of interaction and selective pressure. Antibiotics, the more you use antibiotic, the more you find resistance, hence the antibiotic stewardship. But this is an ongoing battle, and the benefit from antibiotics obviously outweighed the risk of resistance, because we need them and they prove useful. Likewise, vaccination is very important to know. And then as we go along, human being will have exposure and then we will have the antibodies and the immune cells.
Okay. Okay, thank you.
I have a question. Yes,
Yes,
I have a
Dr.
question. Actually, it's
Castro?
yes, I have a two-part question. And I've been in practice as a critical care specialist for 30 years. And one of the things that in my experience is always when we have a new disease, you know, we're rushing to just save the patient, to do whatever we can to save the patient. Then once we figure more or less what to do more or less, then we start looking at the nuances, the little details that sometimes either make the disease worse or make it easier for us to handle the disease. Having said that, two questions, Dr. Khalid and the rest of the panel. There's a lot of research coming out right now about biofilms, especially biofilms in the oral, how that either will precipitate a more severe infection, protect the virus, and then the concept of co-infection, what came first, the chicken or the egg? And as it's been my experience when we deal with biofilms, because besides a critical care specialist, I happen to be a pulmonologist also. Biofilms are very important because antibacterials cannot penetrate. And also they can serve as a way to protect and hide from the immune system. So what Dr. Khalid, starting with you, what do you see and what's your concept about how biofilms are playing in this pandemic?
Well, thank you for this is a very important question. In fact, if you talk about biofilm in bacteriology, any foreign body except the stent in the heart, you will have eventually a biofilm. There's no doubt about it. And this is very well known, you know, Legionella disease. And the example, how do you know there's a biofilm? If you have a hose in the garden and you put your finger, you'll find a slimy material, and that tells you enough about the biofilm. Now, virology. Virology is because of the difficulty of the culture, and now we are equipped with a PCR. So, and we have more antivirals. It's become more interesting. You know, formerly, we don't have antivirals. We don't have a cyclovir. It came recently. And, you know, HIV came, and there was only one antiviral. Now he said plenty, and HIV every day, then change its structure or genetics. And on top of that, we have the PCR. So I asked this question before. What is, you know, we got a normal flora. Now we are testing everything by PCR. It will have a huge data, and then we will start again. What is normal flora, and what is biofilm? And this biofilm, the only bacteria, as we know, and the polysaccharides and all these nice pictures, or there are hidden viruses. And don't forget, I mean, when I was in the Brompton, we had a published paper about the effect of viruses on bacteria, the phages, and it's still not, you know, coming up from the ground, but still an idea. So the answer, I don't know.
Thank you.
Okay, thank you. Thank you so much, Dr. Khalid.
Okay, I have a question.
Okay,
also.
Dr. Fadel, you can ask anyone.
Okay. Merry Christmas again for you all. Hi, Hector. How are you?
Hello, Joseph. How are you?
Okay. I have a simple question. And I think that this question is commonly repeated in TV, in social media, that people vaccinated, people previously infected with COVID-19 can get infection with the new strains. Is there a possibility for those two kinds of people, I mean, vaccinated or previously infected with COVID-19 can get infection with the new strains? Yeah.
I can add another question to this barrage of questions, that those infected with ordinary COVID, are they going to be infected with the new variant?
That's a good question. May I address it?
We don't know. I mean, we didn't answer the question last panel, and still we don't have enough data to answer it. The question, which is, I think somebody, if he tried to challenge me and challenge the vaccinologists, how come that the viral infection itself give you a short-term immunity, but the vaccine give you longer. And we know natural infection give you longer and better immunity. But there is an essential need for the vaccine to boost the immunity. This question has not been answered. So we go along, we fight the virus, and the answer will come later. And I can assure you, science is so much advanced. Sharing of knowledge is so wonderful that nobody will have a disease in the jungle of Australia not known by somebody in South Kent.
Okay, thank you so much, Dr. Khalid. Okay, Dr.
Nathan.
Yes, Dr. Salman.
Let me come very quickly because the last question was really very important.
Okay.
We need to understand, thank you for the two speakers, excellent. We need to understand that this mutation is not fully understood. Although there are 3 variants now circulating around the world. One mentioned in South Africa, one in Asia. Khalid said South Korea managed very well. Actually, the virus now spreading very fast in South Korea and in Japan. And the other one is in England. This mutation is happening and increase the transmissibility of the virus. The transmission of the virus increased by 70 up to 72%, maybe more. So far it replaced the current virus in the UK. And you need to know the UK is the most country in the world which do gene sequencing. The gene sequences in United Kingdom is about 157,000. In United States is the second country in the world, is about 51,000. And the third one, Denmark, is about 16,000. So we have vast of information. And I think our meeting with the House of Common Science Committee recently and with WHO yesterday and group of clinicians from different parts of the world showing 3 things. I mean, I just want you to remember that the company which is manufactured the Pfizer vaccine, the BioNTech, chief executive said, we don't know whether the current vaccine will actually tackle the new variant. He said, we will answer that in 2 weeks. We may need to change, if we need to change, the composition of the vaccine. That could be done in 6 weeks. So the answer, whether the vaccine is effective or not, we don't know. Number one. I think number 2, the vaccine is not antibiotic, by the way. To compare it with antibiotic is like unfair, because antibiotic is direct toward the bacteria or the viruses or fungus or the organism which we are trying to tackle. In this case, with the vaccine, is we are raising the different type of immunity, cellular and fluid immunity, which all of you familiar with, which we don't go through it. Actually, there is a school of thought now, and from the published data by BioNTech, Pfizer didn't publish anything. BioNTech published actually the data now, and it's available for us, showing that the first dose of the vaccine generate about 51% immunity level in the body. But after 3 weeks, i.e. at the time of the recommended second dose, the level of immunity reached 91. So there is a really call from politicians, including the infamous Tony Blair and Professor David Salisbury, well known, the guru of immunization around the world, saying that we don't need a second dose at this stage. This stage is really to increase the coverage. Vaccine is effective, as Seema said quite rightly, and therefore we need to really improve the coverage. I just want to say a couple of things about the new variant. The new variant actually quite risky, although the severity of disease is not recommended, not recorded. We don't know. Until now, there is no record of very severe cases. But the admission to the hospital in the United Kingdom is on the increase. Same things in Japan, same things in South Korea, same things across Europe, where the English variant, the so-called, now spreading across Europe. And that's why government taking a very solid measure. B, that we know that actually the virus affecting young people more. And clinicians in different parts of the world, I agree with Majid, may not be reported in the UK, in different parts of the world reported increased number of children with moderate disease, including colleagues from Basra reported to us yesterday in the meeting that they have a number of cases which are actually now trying to figure all the cases in the Middle East of children admission due to COVID, which they didn't see before. And C, they noticed reinfection. Again, the number of cases of reinfection reported with the previous variant is very low indeed. They are a handful. But now, actually, they reported reinfection. And that's why countries are taking seriously, including the United States now, almost close their border, except with people with negative tests. And by the way, my last point about the test, ordinary PCR are not effective in detecting the virus. You need the 3 gene PCRs. A single gene PCR and lateral quick tests are not actually effective in detecting the new variant. Thank you.
Thank you so much, Dr. Salman.
I have last question. Yeah.
Excuse me, Dr. Abdelhamid, do you have a question?
Yeah.
Okay.
Good day, everybody. Merry Christmas for all. Thank you for the nice presentation. As you know, most of population and government scientists around the globe, they are worried about the vaccine. Does it work now or not? Still, there is a hope to work. But my question, if there is more mutation happen, which is expected to happen, should make us more worried about the vaccine? And my second question, why the mutation appear in some country rather than other countries? Is there a relation with genetics or immunity of the population?
No.
Okay. Who can answer the question?
Yes.
Okay, Dr. Seema, okay, you can answer.
Thank you. Thank you so much for Professor Dr. Salman for the wonderful point presentation just now. Okay, so as for answering this question, first of all, the mutation in particular geographical areas is more economically and politically driven than having to do with immunity or genes. And this is the answer to the second part of the question. And right now, of course, more mutations are expected. However, I am going to put this to a clinical level. So let's say we are, a person gets infected with COVID and the lungs are compromised. Okay, so the person has atypical pneumonia and is being treated, atypical pneumonia as a result of COVID infection and is being treated for that. Other respiratory organs remain untouched. Therefore, there is a possibility for a mutation to affect other organs. Hence, the person being possible to get infected. And those other organs that were not previously infected or affected are going to therefore fall under that category now. In terms of the vaccine, it is said that it is basically treating 90% of all COVID-19 related cases. And from what's going on right now with the mutations seem to only involve the spike protein, which basically means that the transmissibility might be worse, but the actual severity of the disease might not change, which basically means I don't think we should be that worried in that sense. Once again, we have to monitor the tracing and the vaccination. We just need to monitor and change the compositions and the tracks of these two things that I just mentioned. And then we just go from there based on how the circumstances lie.
Okay, thank you so much, Dr. Sima.
Let me come here because this is very vital question.
Okay.
I think Dr. Abdelamir, the so far virologist and Professor Elani from actually University College London and other colleagues in their report to the Science Committee of the British Parliament day before yesterday, they were very specific and very clear that the misuse of antiviral treatment, which WHO recommended not to use, may be one of the major sources of the mutation. I qualify it with maybe. So they definitely all agreed that the changes in the virus is the source of the mutation rather than the behavior of the people. So why the virus changes, it's actually in response to what happened in our bodies, you know. The vaccine, of course, is not a treatment. Vaccine is basically given to healthy people. And we recommend strongly that all healthy people, include those who recover from the virus, should take the opportunity of having the vaccine. Once the mutations are detected and reported, before I answer your second question, the changes of the vaccine is very easy, as we've seen, because the technology is available, as we've seen with the flu. The flu vaccine we change every year. Last year vaccine component is different than this year vaccine components because we add whatever mutation happened in the coronaviruses or the flu. The last point you said, is it happening in specific countries, not other countries? We don't know. To be very honest with you, simply because of gene sequences and the ability of each country to detect changes in the viruses. Iran, for example, they reported the same strain which exists in United Kingdom. Although there is no flight between the two countries. So we don't know whether this is actually changes in the virus happening in Iran. But Iran have gene sequencing ability. Other countries, they don't have. As I said, the United States is about 5 times bigger than United Kingdom. Yet they have very, very low capacity not capacity, but or willingness to do gene sequences, which is really shame, you know. And I'm sure if they do the extensive gene sequences, they will discover the same variant or even different variants in the United States.
Okay, thank you so much, Dr. Salman.
Can I ask one question?
Yes, Dr. Majid.
I would like to ask Professor Salman and also to the opinion of Dr. Abdelamir, since they referred to increased cases in children in Basra, as among other countries, Professor Salman, is this a new surge in cases, because I'm in contact with other colleagues in Suleimania and Erbil and things, and they have number of children as well, but it was related to the original, if you like, variants. And if so, if this surge is high, is it? Are they doing the genetic sequences in Iraq? Have they detected the mutation? Thanks.
No, they don't have. Iraq, they don't have gene sequencing facilities at all. Most of the IMRO region, they don't have that except Iran. Yeah. I don't know whether Egypt has such facilities or not. So far, we received from WHO IMRO information, actually a couple of emails today exchanged, because we are trying very hard to provide Iraq with facilities for gene sequences. And I think the minister last night in our meeting of the COVID committee agreed that he will fund 2 machine, reagent, and software. It's a very complex process, and you need to train a good number of people. For your first part of the question, Ali Rahim from Basra, which you know, I'm sure, reported new cases, which he never seen before, and he reported reinfection. Iraq collect data on reinfection, and Dr. Riyad yesterday instructed all the hospitals to provide data on reinfection. So hopefully we will know about it today, tomorrow.
Thank you. Probably it is too early also for UK, because especially now the Royal College in recession of pediatrics.
Okay, thank you so much. Any question?
Yeah, last question.
I have a comment.
Sure.
Yes, Dr. Fadel, yes, you have questions?
Yeah, I have last question. I want to ask Dr. Seema.
Yes.
Even on theoretical level, vaccine can be blamed to be a cause for emerging a new variant or new strain of COVID?
No.
Okay.
No, absolutely.
No, there is no evidence. This is only a conspiracy theory. And right now is finally the time for us to totally eliminate conspiracy theories and work based on scientific information and knowledge.
I think I 100% agree with Dr. Seema, because the mutation, the current 6970 mutation in the UK happened in September, long before the vaccine.
Yes, exactly.
Number one. And secondly, it's covered 72% of the population in Southeast England, you know, yet in Southeast, the entire country, we managed to immunize only 800,000 by tonight, you know. So,
yes.
So all this about linking the mutation to the vaccine is nothing but rubbish. And we have to say rubbish. Are you?
I just like to add one comment.
Yes,
that Dr.
Salman.
Dr. Salman pointed to the point. Unfortunately, we're going to have to wait until mid-January for the United States to go 100% to use all its technology capacity, because for the past year, the administration here in the United States has not been committed to that. So that's number one. So unfortunately, we will be getting to that point. But also from a clinical perspective, we have to be careful, because the tendency in clinical medicine, and this sometimes is driven by trying to create guidelines or sometimes being more cost effective, that we start saying, this is how we're going to handle certain patients. This is how we're going to treat them. And this is a rapidly evolving disease process, and also a rapidly evolving capability, because we're talking to people from all over the world. And as Dr. Salman is mentioning, that some people have certain capabilities or not technologically wise. So we have to understand that if we're seeing a failure, or if we're seeing a manifestations of new mutations, the question really is that because the patient was really not afforded the best treatment from a clinical point of view? Because when we talk about a mutation, and maybe Dr. Khalid can answer this, when you talk about a mutation, just having a mutation doesn't mean you're going to get a clinical disease, because then you have to talk about inoculation dose. How much viremia do you have to get a clinical manifestation?
Okay.
Well, I mean, I cannot answer this question categorically because viremia detection, so far we do PCR, which is a qualitative, not a quantitative DNA test like what we do with CMV or EBV or HIV or even hepatitis. So that is one thing. I agree that there is a rush to do everything to the patient who are dying by giving them any antiviral medication, which could well be, has been indicated, to be the cause of mutation. There is another thing. There was a paper about saying that one cause of mutation could be the plasmid given to the immunocompromised that may help to create mutation. And the other thing about the surveillance. Now, let's not forget, because we are much better in surveillance and trace and detection in the last 3 months or 2 months. What about the previous months? So the picture is not quite clear. However, the domination of the new strain is actually a serious thing and is alarming. But I mean, we are waiting for that. Having said that, it is very unusual for too much mutation in the virus to happen now compared to the previous months. And I mean, initial literature saying that COVID does not accumulate mutation compared to, flu viruses. However, this is a new thing that we have to watch. And as has been said, it could be that we are having a different clinical picture of COVID compared to the earliest one.
Okay. In 1 minute, I want answer about this question. Is this new strain was expected? And in the worst scenario, more strains can be emerging. I want answer about this during this webinar.
I think Dr. Khalid said
it depends on it.
A mutation happening in all type of viruses. But is it expected? I doubt it very much. But I think, let me finish this with some positive remark. If you look at the virology of the new strain, the way virologists describe it, the new variant actually added about 6 spikes to itself, But at the same time, which is the good news, this is the bad news. The good news is deleted 2 spikes.
Yes.
So as far as the virus start deleting some of these spikes, as we've seen in SARS-1, for example.
Defect virus.
It started to go inert in no time. So scientists now looking at these positive things, hopefully the next mutation will delete more spikes and the whole thing is over. We don't know. God only knows.
Okay. Thank you so much. Thank you. It was a really very informative talk and lecture to improve our knowledge about last update on COVID-19. We will meet you next webinar with a new trending subject. See you and stay safe, please, and Happy New Year.
Merry Christmas.
Thank you. Happy holidays.
Happy Christmas and Happy New Year to all of you.
Thank you. Bye now.